Generalizing genetic predisposition of cancer risks.
I mean, to this wonderful workshop. The quality of the science has been amazing, and I've been really enjoying my first trip to India, and I'm sure I'll be back sometime. Yeah, so today I'm going to talk to you about some of the challenges we're facing when it comes to globalizing or generalizing cancer predictions across the world. And then the first half of the talk is going to talk about the challenges, and then in the second half I'm going to focus on a case study which is looking at prostate cancer risk in Africa. So when you look across the globe, what's really interesting is when you look at a different country, you find that the primary killer when it comes to cancer really varies. So this is global CAN data. And on the top part of the figure, we see what are the cancers that tend to be the most important for men. And then for women, it's on the bottom. And this is once again looking at mortality. And so when you look at men, you tend to see that lung cancer tends to be the primary killer. But if you look at sub-Saharan Africa, prostate cancer really jumps out. And here in India, we can basically see that lip and oral cancer actually plays a huge role when it comes to mortality. We look at across the globe, we see different patterns for women. So in some countries, once again, it's lung cancer. But in other places, it's breast cancer, or in sub-Saharan Africa, a lot of times it's cervical cancer. And so one question that comes up is, well, why are different cancers more common in one part of the world as opposed to another? Or why are they more lethal in one part of the world as opposed to another? And so one thing that contributes to this is the fact that cancers are genetic diseases, okay? I mean, there's lots of things that affect cancer. We think about all these mutations that can contribute. So this is a classic figure. It's showing, the traditional hallmarks of cancer. But the idea is there's lots of ways that tumor cells can have mutations that actually makes them more fit, and they can evolve to be very harmful to us. And so the idea there is that we can have different numbers of mutations in our germline or in somatic cells, and we can think about that in terms of a disease burden. And the idea is that some of our genomes carry more deleterious mutations. So in the United States, we might use a baseball metaphor and say somebody was born with 2 strikes against them. Here we maybe have to use a cricket metaphor, and that would be like basically a team that basically starts a match a few wickets down. So it starts with a disadvantage, okay? So the idea is that we all don't have the same amount of these deleterious mutations, and the idea is if we could quantify this, maybe we could triage individuals in terms of risk. So one thing that Kane Patel in my lab did is he looked at a huge number of GWAS loci and basically said, okay, what's the risk allele frequency in Africa, and what's the risk allele frequency outside of Africa? And to me, the boring ones are the ones that are along this line, where it's identical across the world. Okay, what's really interesting are places where maybe the risk allele frequency is at, say, 60% in Africa or 10% outside of Africa. If you think back 2 talks, Wei mentioned a deletion that happens to be much more common in East Asia, and it's basically absent from Africa. It's the same sort of thing. And then the real question is, how do you get these differences? How do you have these big differences in allele frequency for these loci that are associated with major diseases or cancer? And so there's lots of evolutionary reasons why this can happen. I'm not sure why of decided to go to the left, right? But it's there. Okay? Yeah. So first of all, you can think about how natural selection can contribute to this. Okay, so one way of thinking about this is you can think about, well, there could be a selective sweep or partial selective sweep. Maybe one of these mutations happens to be evolutionary beneficial, and that's one reason why you find it in one part of the world. Or maybe it's linked to something that matters. So that's definitely one way that you can get these big differences. Another way that you can actually have it is when you have population bottlenecks. So you have this sampling of human diversity as we've expanded to discover new regions of the world. And that can lead to these big allele frequencies too. The third one is actually less common, but it still can matter. So most of the people in this room have basically 1.2% Neanderthal DNA, and we all have different Neanderthal DNA, and then sometimes that DNA can actually be beneficial or it could be deleterious. So the fact that we've had these mixing of populations, even with guys that, well, if all Neanderthals look like that, we'd probably have more than 1.2% Neanderthal DNA. But the idea is that we have these mixtures of genomes, and because we've got these mixtures, some of us might have more or less mutations that contribute to cancer. So one example from my lab was we actually looked at prostate cancer in Africa, and we zoomed in on a number of genomic loci. But one actually that was really interesting was on chromosome 2, and there's this entire region here that's associated with prostate cancer. And what we did is we looked at signatures of positive selection, so that's what this y-axis is here, and there's this huge haplotype that basically looks like it's beneficial in Europe. And what ends up happening is this haplotype also contains a protective allele against prostate cancer. It doesn't have a big odds ratio, but it contributes to the disease. And so it turns out that there's a couple genes in this region. one of which is PRLH, so that's prolactin-releasing hormone, okay? And you can see that, well, that might make sense why that might be involved in prostate cancer, but doesn't explain why that might be under selection. Well, right next to it is melanophilin. And you go, well, what does that do? Well, if you have that same mutation in a dog, you get something that looks like that. And it bases this diluted pigmentation. And so what appears to have happened is that there was selection for pigmentation in Europe, and it just happened to drag along an allele that was slightly protective against prostate cancer. So this is one way that you can get allele frequencies that look like this. So the blue here is the risk allele for prostate cancer at this locus. So this is one way that you can have evolutionary history that can contribute to differences in disease risk. So given that we've got these big differences in allele frequencies that can occur, the question is, how do you find them? And so we've had, a number of talks and mentioned genome-wide association studies. Here shows some data from the UK Biobank. Shows it for prostate cancer. And you might notice a huge peak on chromosome 8. That's an 8q24. Makes sense. It's near MYC. Okay, classic prostate cancer locus. If you do a study in Europe, that's going to come out. It turns out if you do a study in Africa, it's probably not going to come out as strong. And one big difference is that the risk alleles there tend to be much, much rarer. Or actually, it's more common, sorry. But you don't find them at 50% frequency. Okay, so one thing to keep in mind is that when you're doing a GWAS, your statistical power really depends on the allele frequency and the effect size. So if a mutation happens to be monomorphic in the population you're looking at, you're not going to catch it. So what we know right now about cancer genetics, it's really biased. Okay, it's biased in terms of where these studies were done. It's biased by how big these studies were done. But given that we still have a huge number of cancer studies, these are not showing unique associations, but given that there's been 600 plus cancer studies, we have this huge number of these associations that are there. And so we can use those to build what are known as polygenic risk scores. And the idea here is we're basically counting up the number of deleterious mutations in somebody's genome and is giving some way of a quantitative way of describing risk. Okay, and it's actually a really, really simple way that people tend to do this right now. I mean, if you look at the equation, you're like, wait, that's way too simple to describe the biology. And the truth is, you're right. Okay, so the idea here is that you select a number of SNPs that are associated with the cancer that you care about. Okay, you might use just GWAS data, you prune a threshold, and then you basically do LD pruning. Or you can use machine learning, but once again, for that, you run some risk of overfitting. The idea here is that you look through somebody's genome and you look at each locus. First of all, you count how many copies of the risk allele they have. Do they have 0, 1, or 2? Then you weight that locus by how big of an effect it has, and you just sum across the genome. And the idea is that some individuals are going to have a higher polygenic risk score, some are going to have a lower risk score. And what's really useful is you can use this to triage risk. Okay, so this is a recent paper that came out in Nature Genetics a couple of years ago now. And so what Carrot al did was they looked at a number of traits, but this is showing for breast cancer. And what this is showing is the prevalence of breast cancer based on what percentile an individual is when it comes to their polygenic risk score. And you can see this, that if a woman's in, say, the 99th percentile of her polygenic risk score, she's at least 4 times more likely to get breast cancer than somebody that's on the low end. Okay, and you can see there's some subtle effects, but you really don't want to be on the high end of the curve. And where this can be useful is if you're trying to decide, okay, what age should somebody go in for mammogram screening? Okay, or how often should somebody be screened? If somebody's in the high end of the curve there, then maybe somebody should start earlier. Okay, it doesn't say you're going to get the disease, yes or no, but it still is informative in that sense. Okay, and so there's plenty of utility for this, but there's also a risk that as we build these scores, they can be overhyped. And so there's a lot of reasons when we're building these that you have to be really, really careful. Okay, and first of all, there's this idea of the missing heritability. So a lot of genetic variants that contribute to cancer, we just haven't discovered them yet. Okay, we haven't had well-powered GWAS that can catch all of these. Okay, so we only currently know a small fraction of the genetic variation that contributes to these diseases. The other thing is our polygenic risk scores, they have a really, really simple genotype to phenotype map. It's really sad because we're basically ignoring everything we know about cell biology. And so if we could incorporate that into our equations, we could do a much better job. Also, the idea is that these polygenic risk scores, they actually work a lot better to say, okay, you're low risk. Okay, if you're trying to say you're going to get the disease, a polygenic risk score is not going to work that well. So in terms of statistics, you can think of the negative predictive value is much higher than the positive predicted value. So these are general limitations of polygenic risk scores for everything. But we can also think of some other things that could contribute to some of these challenges. And so one thing is the problem that almost all of our GWAS have used samples of European descent. So this shows the number of individuals that have been in genome-wide association studies over time. And you see, well, we're doing a lot of GWAS, but way too many of these GWAS focus on individuals of European descent. On the right, you see what the proportions are in the world, and the world is not European, okay? So if we want to basically catch these risk alleles and use this to predict disease risk, we need to do more GWAS in places like India, or places like Africa, or places like China. We need to look all across the globe, because that's where the world's population happens to be. Another thing that could contribute to some of these challenges is that when we do a GWAS, we tend to use a genotyping array. I mean, it'd be great if we could use whole genome sequencing, 30x coverage, but even the NIH doesn't have that level of money to do it on a massive scale. And so we use these genotyping arrays, and these are great tools, but they also provide a lot of challenge, because how do you decide what SNPs get on the array? And so a lot of the SNPs that are used on these arrays are ascertained in European populations. They're intermediate frequency in these populations. So if you use these to do a GWAS, well, guess what's going to happen? You're going to catch variants that are intermediate frequency in Europe, but you're going to miss the ones that happen to have different frequencies. So the technology used can also contribute to this. Another thing to keep in mind is that if you do a GWAS in one population, it might not replicate in another. Okay, so you could do a study in Europe, you could say, well, would it work in sub-Saharan Africa? Would it work here? And one way to think about that is that you could have an allele that tags risk in one population, maybe it doesn't tag in another. And that could be due to linkage disequilibrium, it could be due to lots of reasons. Another thing is you can have effect sizes that vary among populations. Maybe the odds ratio is 1.4 over in China, but here the odds ratio is 1.2. And the question is, well, what could be doing that? You know, maybe there's some sort of epistasis going on, but it could also be a genotype by environment interaction. So when we think about things like that, you can even just think about when we're trying to predict genetic risks of cancer, we can't ignore the environment. So this is old data. It shows the proportion of individuals that are smoking in India way back in '98, and you can see how much it really varies across the country. If you were to try to repeat this study now, I'm sure you would see very, very different numbers, because policies have changed. But the idea is that if somebody's at high risk for lung cancer, you really want to know, well, are they smoking? Are they in a region with a lot of pollution? All these things contribute to it. So if you just look at genetics, you're only going to have a tiny, tiny bit of the picture. Lastly, what I want to mention about cancer predictions is that there are very, very different things that happen when we focus on cancer. And so I think it was the first day of the talk, there was a number of points that said, well, let's focus on somatic mutations. And most of what I'm talking about is focused on germline. But when we think about cancer, yeah, we can predict based on somebody's germline, but then all throughout your life, you're going to be getting these mutations. And then how do we predict that? You know, so if we're trying to predict cancer risk, maybe we need to screen those somatic mutations somehow. Maybe look for circulating tumor DNA in somebody's blood. The question is, how do you even catch that? But it turns out, and this is actually a little bit surprising, is that a lot of cancers actually have high heritability. So prostate cancer's heritability is about 54%. Also, when you look at cancer versus non-cancer risk scores, they work equally well, or you could say they work equally poorly, but they all work about the same. So even though somatic mutations matter, germline mutations still are important. Also, you could have different types of cancers can mutations in different genes. And then there's actually a really interesting point here is that Kelly Harris at the University of Washington was looking at germline mutations, and she actually found trinucleotide repeats, or not repeats, but trinucleotide motifs, which actually have different mutation rates in different continental populations. So we all have mutations. You think 1.2 times 10 to the minus 8 per base pair per generation. But at different motifs, we might have different chances of mutation. So different populations can have subtly different mutation rates. And all of these are going to contribute to making it more challenging to predict risk of cancer. So one way to look at this is to focus on prostate cancer. And the reason that my lab does this is that prostate cancer has a very big difference in both incidence and mortality throughout the globe. So we can look at data from the World Health Organization, and we can basically look across the entire Earth, and we see in sub-Saharan Africa and in the Caribbean, we see that there's much higher mortality of prostate cancer. I don't know what's going on in Turkey, but it's elevated there as well. Okay? So we see it where it's not monomorphic. There's a lot of variety there. We can even look within a country. So this is showing the Southeast United States, and in this panel right here, we see prostate cancer mortality. So you can see it's very blotchy. You can see there's some parts of the United States where it's much more common, others where it's pretty rare. And then this shows actually the proportion of individuals that are African American in that same region. And you can see there's this really high concordance between these two. And that suggests that maybe there could be a genetic component. But you might also be wondering, well, could this just be access to care? Okay? If that was the case, you might see this for every cancer when it came to an African American. But what happens is if you were to look at the same sort of map for something like testicular cancer, you wouldn't see this concordance. So something's going on there. Healthcare quality also matters. So I'm in Atlanta, which actually has a lot of African Americans. It's right here. But actually our prostate cancer mortality is pretty low. So some of it has to do with hospital care as well. So given that, what ended up happening is I joined this group called the MADCAP Network, and it's led by Tim Rubbick. And what we're doing is we're focusing on prostate cancer in sub-Saharan Africa. So we're teaming up with a number of study sites in Africa. And what we're trying to do is see, can we discover novel variants in Africa? And can we also predict these risks using what we know right now? And so one thing when we were doing this study is we realized that if we wanted to do a GWAS in Africa, we were going to have some challenges. So we're accruing roughly 6,000 to 7,000 individuals, and what we're trying to do is we're trying to find the genetic variants in Africa that are associated with this disease. But if we use any off-the-shelf array, we're going to have some problems. So if we use the H3 Africa array, it's not going to be great for cancer studies. If we use the OncoArray array, it's not going to be great for African populations. So what we ended up doing is we designed a custom genotyping array that works well for the study populations we're looking at and for the disease that we care about, which is prostate cancer. The array actually will work pretty well for other cancers, but it's optimized for this one disease. And so we worked with CPGR and also worked with Thermo Fisher to help build this array. And what I'm going to talk about over the next few slides is our preliminary data that comes out of this. And what we did is we looked at a pilot data set of 399 African cases and 403 African controls. And so just to give you a little idea of what our array does, is this shows, the Venn diagram shows how many of our 1.5 million variants overlap with the H3 Africa array, and how many overlap with the OncoArray array. And we purposely chose genetic variants that happen to be on other arrays. But we also included a lot of genetic variants that happen to be EQTLs in the prostate, because if something could affect gene expression in the prostate, it might make sense that that could be involved in prostate cancer. This shows the density of markers when we look at AQ24, so mix over here, and CCAT is PCAT1 and PCAT2 are here. And so this region you think about, it's very important for cancer risk. If you look at the OncoArray array, which are these gray dots, basically through most of this region what we find is that there aren't that many markers on their array, but when it comes to this region, AQ24, OncoArray array is great. H3 Africa array, it's okay across the entire genome, and ours basically tries to combine the strengths of both of those. So using this array, we looked at African individuals from 7 study sites. So looking at 1 study site in Senegal, we're looking at 2 from Accra, Ghana, 2 different locations in Nigeria, and 2 in South Africa. For each one of these sites, our pilot data set has roughly 59 cases and 59 controls. So you pool it all together, and it's roughly 400 and 400. This is showing an admixture or structure plot, and it's looking at it in terms of country, in terms of study site, in terms of case control status. And the one take home from this is that whenever we look at a study site, we see that our cases and controls tend to have the same genetic ancestry. There's some subtle differences, but in general, we don't see a huge difference within each location of saying, oh, the cases must have more Khoisan ancestry or Zulu ancestry. We don't see anything that jumps out like that. We can also identify, using this array, individual loci that have big allele frequency differences within Africa. So actually, if you think about androgen receptor, it can have important things with respect to breast cancer. It matters with respect to prostate cancer. So what Melanie in my lab did was she looked at the androgen receptor gene in Africa, and there's actually a risk factor that's close to the gene that's associated with prostate cancer. It turns out that the risk allele is actually much more common in South Africa than it is in West Africa. So by using this array, we were able to identify that there's a lot of heterogeneity within the continent when it comes to allele frequencies of loci like this. But most importantly is what we wanted to do is we wanted to use this array and use this study population to basically say, okay, we know that there's prostate cancer in half of our sample, the other half happen to be controls. The question is, can we use an existing polygenic risk score and say, well, can we predict the guys that are going to get prostate cancer? Can we predict the guys that don't have it? And so what we did is we used the best GWAS for prostate cancer to date. So it's the Schumacher study that was in Nature Genetics in 2018. And what they did is they identified a large number of loci. It basically was a little bit less than 150 loci. There's independent loci. They're associated with prostate cancer. We took that. We used that data to build a polygenic risk score. So basically, we took what they had and did a few slight changes. And then we applied that to two data sets. We applied that to our MADCAP Africa data set. That's the blue here. We also applied it to the UK Biobank. And when we applied it to UK Biobank, we'd exclude a number of populations because we basically didn't want to have this heterogeneous population. So we only looked at Europeans. I don't want to say, well, everybody's European if you're in the UK, but basically white Europeans in the UK. We excluded individuals of recent African descent or recent Asian descent from that. And then what we did was we basically said, well, what are the polygenic risk scores for prostate cancer in both these data sets? And so just to give you an outline here what's going on, when we look at the solid lines happen to be controls, the dashed lines happen to be cases, and once again, black is from the UK Biobank, and then the blue color is in the MADCAP. And the first thing that jumps out here is you can see all the black are shifted left, and the right happens to be blue. And what that means is that if you were just to blindly look at a polygenic risk score, if somebody was African, you would most likely say, oh no, he's likely to get prostate cancer. So what seems to be the case is these polygenic risk scores are massively elevated for African genomes. And that's independent of whether they're a case or a control. Okay? And one of the problems with this is that those SNPs that are used on this polygenic risk score, they didn't come from African populations. They came from European populations, and so they're carrying that bias with them. Okay? Ideally, you would want both of these curves to be sort of shifted to be much more overlapping. Okay? If there are real differences in risk between these populations, then they're not going to be exactly overlapping. But the fact that there's such a shift to the right when it comes to the blue curve suggests that something's going on in terms of this polygenic risk score, and it's not going to work that well in Africa. The thing is, when we look at both these data sets, we see the shift in terms of the cases versus the controls. So it turns out that prostate cancer cases do have higher polygenic risk scores than the controls. But there's plenty of individuals that happen to have a high risk score that are cases, that are controls, and plenty of ones that have a low score that are cases. So we can see the separation here is actually sort of saying, well, it's somewhat informative. And this is where it gets a little bit depressing, is you can look at these ROC curves and you can basically see, okay, how well does this actually predict? And this is currently our cutting-edge prostate cancer polygenic risk score. The AUC in Europe is only 0.67. Okay, it actually doesn't work that well. When you try to apply the same polygenic risk score to Africa, it does even worse. I mean, if it was by chance, you'd see this blue line right here, and you'd have an AUC of 0.5. So it's informative, but it's not that great. Okay, and what this means is that if we're trying to predict the risk of this particular cancer or other cancers in Africa, we need to have polygenic risk scores that are generated from populations that have very, very similar ancestries. You can't just take something off the shelf from Europe and translate to Africa. Whether it would translate well to India is an open question. It might work a little bit better, but how well, we don't know yet. So going forward, when we think about, well, what does the future hold? Well, the first thing is we need to have caution when we're using polygenic risk scores. Okay, that doesn't mean just to say, well, just ignore them, they don't work well. But it means that when you see them, you have to be very careful if you're applying a polygenic risk score in terms of both the ancestry of the original samples and also the ancestry of where you're trying to apply that. Okay? The other thing is, partially because of that, there's a major advantage in performing GWAS across the globe, okay? And that's one reason why we're studying in Africa. That's why a lot of the people in this room are really interested about genetics in very interesting places. And then finally, because we know that there's these biases, there actually are ways to correct for all these problems, okay? So some of it is, if you know the bias that a particular genotyping technology brings, maybe you could correct the risk score for that. Or if you know the bias that happens if you happen to do a study in Europe and try to translate that to India, maybe you could do some sort of correction. One thing I'm not describing in this talk, we've actually found that knowing some additional evolutionary information, so knowing if an allele is ancestral derived, that actually can be used to help improve these corrections. So with that, I would like to thank everybody for listening, and especially like to thank my lab, my collaborators, and definitely the host and funding. And once again, thank you.
Thank you, Joseph.